Assalamualaikum warahmatullahi wabarakatuh. I beg to Dr. Hanis and Dr. Leila. I am N.Izzati, binti Mokhzani, matric number 1171483. Today I will present to you my proposal with title Identification of 3 duckweed species and their interaction with Lysinibacillus sphaericus. This is the outline. Starting with introduction, duckweed is one of aquatic plant that belong to family Limnaceae and up to 37 different species that can be found. Duckweed size between 1 to 15 cm. Genera of duckweed are Lemna, Spirudella, Wolffia and Wolffia. Duckweed grow rapidly in suitable aquatic environment with adequate sunlight and nutrient. Duckweed contain high in nutrient. For Lysinibacillus sphaericus bacteria is belong to family Bacillacea. Act as biopesticide. This bacteria is first bacteria that against mosquito larvae in 1960s and also function as plant nutrient enhancer that can enhance germination of seed and promote shoot length. By Zakira et al. 2017. The aim of this research is to identify 3 duckweed species and their interaction toward Lysinibacillus sphaericus in terms of growth rate, plant morphology, and number of mosquito larvae. Continue with problem statement. First, to identify the species of duckweed become complicated due to small size of duckweed. Second, based on Breda Pest Management 2017, watery area will attract mosquito to grow and live there. To avoid it, Sphaericus can be used as biopesticide. Finally, besides function as biopesticide, Sphaericus also can act as plant growth promoting bacteria that can enhance the growth of plant. For research objective, first, to identify 3 species of duckweed by using molecular identification method. Second, to determine the number of mosquito larvae and growth rate of 3 duckweed species in growth medium with Sphaericus. And number 3, to analyze the microscopic observation of the interaction between 3 duckweed species and Sphaericus. For hypothesis, the plant gene for 3 duckweed species could be identified by using molecular identification method. Second, the presence of Sphaericus in growth medium could decrease the number of mosquito larvae and increase the growth rate of duckweed species. And number 3, there will be interaction between duckweed species and Sphaericus, but it will not affect the growth system of the duckweed species. For research question, first, what is the duckweed species? Second, how did Sphaericus affect the number of mosquito larvae and growth rate of the 3 duckweed species? And number 3, is there any interaction between duckweed and Sphaericus? Continue with literature review. Duckweed and Lysinibacillus sphaericus. First, protein content and fat content are found low in Lemna punctata while high in Wolffia hyaline. For starch content is found low in Lemna minor and high in Lemna punctata. For Sphaericus is one of entomopathogenic bacteria that found in early 1990s and act as biopesticide. And another one, Sphaericus bacteria, can be used as plant growth promoting bacteria. For method use, first, identifying of candida auris strain done in short time when use Kanika kit when extract DNA. Second, RPA can detect pathogen-specific DNA or RNA under 20 minutes without using complex instrument. And the third, sample can be illuminated through lens with high source of energy when used fluorescent microscopy. Continue with method and procedure. Material that will be used is 3 duckweed species: Lysinibacillus sphaericus bacteria, mosquito larva, nitrogen liquid, Kanika kit, and NA medium. For amplifying gene using RPA, first grind duckweed by using liquid nitrogen to extract the plant tissue, then undergo the DNA extraction by using Kanika kit, then amplify the DNA by using RPA, then send for DNA sequencing. For the primer, we use RBCL and tRNK MATK. For growth rate of duckweed, Sphaericus culture will be dissolved with growth medium and pour the medium into container B and C. Container A will act as control that contain duckweed species and 3 compost and distilled water. Same goes to container B and C. Container B will be added with Sphaericus culture and container C will be added with Sphaericus culture and 5 number of mosquito larvae. And after 3 days, we need to observe the number of duckweed plant and the number of mosquito larvae. For For microscopic analysis, first, prepare MS media. Second, growth duckweed in culture plate 6 row. The upper row will act as negative control, absent L. Sphaericus culture. The lower row will act as positive control, present with L. Sphaericus culture, and the observation of interaction between duckweed and L. Sphaericus will need to be done after 1 day. For expected result, for amplifying gene using RPA, we expect to see clear band of DNA for each sample of duckweed when analyzed in gel electrophoresis. We also expect to know the DNA sequence for 3 species of duckweed. For growth rate of duckweed species, in container A, we expect to see the duckweed can grow healthier with no interfere. Container B, duckweed can grow healthier although there is presence of Sphaericus culture, and container C, we expect to see the duckweed grow healthier and also the number of mosquito decrease since there is presence of Sphaericus culture. For microscopic analysis, we expect to see in root system of 3 species of duckweed does not contain Sphaericus bacterium, and we also expect to get clear image of root system of the duckweed using microscope. This is my gadget. Since we're still in pandemic COVID-19 situation, we estimate to start the experiment in October. That's all for my proposal defense presentation. Thank you.